welcome to our class about robustness. Let's all start by taking a nice deep sigh of relief. We're here. We're kind of celebrating finishing the first part of the course. The first part of the course is dedicated to the principle of network motifs. The idea that complex biological networks, which would be ununderstandable to the human mind, are made up of simpler circuits that occur again and again and again. Evolution converges independently on these circuits again and again and again. Each one takes a nice deep breath. Each one has its own information processing function that we can understand, and together they build up larger circuits that we can understand based on the smaller circuits. This is another deep sigh of relief. Welcome. And today we'll move to a new part. It's called robustness, dedicated to another important principle that I'll explain. And then in the end, we'll have a third part about optimality. So just as a kind of a review of what we did in the first part, I wanted to show you this music video by Acapella Science about basically about gene regulation networks, right, and what they can do. How many of you know this Beatles song? It's from Bistability.
Hi. Once they'll divide and decide on 1,000 fates. Did you ever figure how they know? Be math. We are built of modules combined in a planned out way. Each new piece must be told where to go. Oh, now there's a science helping us to understand how our cells encode this architectural plan, signaling each other with genetic tools. Oh yeah, wow. Phenotype the interface for mouse and man, genotype the files and the sub-programs. What then are the switches, circuit boards, and boot code? Looking at the logic in the ways that we grow. Every gene directed by a signal key code. Proteins that can activate, enhance, or veto. The signals are controlled by other genes that signal, calculating in a network labyrinth, though, where the heart and liver and the hands and feet go. Signal mapping tells each region what it ought to be, yo. With circuits so deeply built upon, they're older than the Paleozoic era, baby. In a crucial pathway, changes tend to get torpedoed. Where they go, calamity goes. As the cycle shapes, see down, they cascade like a domino, like you and I, Drosophila. The path that makes us optical was laid a long time ago, back before we blew up the Cambrian like a bomb. Now my eye protein can make you see out of your bomb. And Hedgehog and its relatives like Indian and Sonic. Set up in the gradient on segments embryonic. Split for brains and asymmetric parts depend upon it. Flipping on genetic switches and logic from Devo to Evo, adult and embryo. Mostly don't evolve in the genes of the genome. Save for the mutation in the regulation. Keep the building blocks and swap their activation from Devo to Evo. Parts of alter egos, homologs evolve from repeats in the schema. Switch a couple bases in the proper places. You'll be watching flies grow legs out of their faces. Oh yeah, Evo, Devo. Stick around for modern synthesis, the sequel. Only by combining can a new theory grow. Evolution and development, amigos. Diva. Signals trigger patterns of complexity, so switching up the switches of a signaling node gives a modular and simple way to evolve. Switches. Look at our spinal sigma generate an e-row, built on a molecular clock. One circle, one vertebra, baby. Speeding up its rate is snakes, developmental cheat code. Better where a lizard's feet grow. They turn up this amino. Devo. This is how we go from single cells to people. Every generation and in life primeval. Life in variations endless and beautiful. From Devo to Evo, larva to mosquito. Patterns of result as the signals proceed. Never the gene with the glow tag. Kill it with the morpholino. Shortly go morpholino. From Devo to Evo, voyage of the beagle. Body plans evolve when proteins to the genome. Better life's beauty grows. Aesthetic and vivo. Evo, Devo.
This song celebrates, I think it's an amazing scientific revolution in the 20th century, 21st century, understanding of combining ideas of evolution, the deep history of life, the logic of gene regulatory networks and how they make cells think and animal shapes form. And you saw all those activation and repression signs and input logic signs, just to hint that there's a lot to already known and a lot to understand about the really miraculous thing we're talking about, which is the formation of living organisms like people. Yeah? It's quite amazing. And this idea of network motifs is the conceptual, let's say, ability that helps us take these very complex networks and figure out how they work. Right? And we're going to, so that was our musical review. I wonder if we can turn off the projector now. And we want to switch now to look at a little bit different kind of biological networks and look at them from an angle of a central puzzle, you can say, which is the following. When we have biological circuits, they're made out of proteins. And as we'll see, these proteins can change each other, work on each other in many different ways. That's the science of biochemistry, how proteins can modify each other, and together they make computations. And it's essential, in some cases, that these computations be precise. The problem is that we're trying to make precise computations with noisy hardware. And when I say noisy, I mean there's different kinds of noise. In particular, in this class, we'll work on this problem. The protein concentrations in the cell are variable. So if you try to make 1,000 copies of a protein in a cell, one cell will have 800, another cell will have 1,200. You can't get around that. There'll be tens of % variation in different proteins. You can put on negative autoregulation and stabilize it a little bit, but still have variation. So how can you make very precise computations with noisy hardware? Of course, this question has occupied engineers for, since there were engineers. When you want to make a steam engine, you want to regulate it, so you build these regulatory things, that spin. The more steam is there, and they pull out the little iron balls, and you get this kind of regulator that opens up the steam valve. And in electronics, you want to make circuits that don't depend too much on the inaccuracy of the resistors and capacitors you have there, because you can't have absolute precision, and still you want them to work. Or when you synthesize a cell phone, you're guaranteed that the silicon won't be exactly like you want it, and still you want it to work. So a lot of engineering is robustness, is how to design circuits to overcome the most common source of variation. So the principle of robustness is that biological circuits have evolved to have a structure, usually a beautiful and quite cool structure, that makes their essential function insensitive to naturally occurring variations or fluctuations. And so we have to know what function we're talking about, robustness of what function we're talking about, and what variation. Robustness of what with respect to what. So in our body, we'll talk about how insulin secreted by the pancreas is robust to changes in what happens in organs far, far away, like muscles, etc., that you have no idea what they do, but you still want to work this no matter what their properties are. So that's one kind of naturally occurring variation. We'll talk about that in another lecture. But today we'll talk about a function of signaling proteins with respect to protein variations inside a cell. Right, so this is a general question. Any questions so far I should ask? About part 1 of the course? Maybe I'll stop till there's a question. Take a nice deep breath. Welcome. Yeah.
We were talking last time when you do the bimodular that you want.
Bistability.
Bistability.
Yeah.
And we were talking about why you have bias against circuits that will oscillate.
Yeah.
But if you have like a pathological situation like cancer, do you see the patterns of like oscillation? Maybe it's beneficial for those kind of systems?
So your question is, if I understand it, we talked about bistability, where there was a mutual a positive loop made of 2 transcription interactions. And we said that a mutual loop of a positive and a negative transcription interaction, this is prone to kind of noisy oscillations. Yeah. And we saw that the double positive, for example, that gives you this bistability situation where with memory, etc., this bistability. And this occurs in developmental networks, like what we saw in the movie. You want to remember that the tail of the snake stays the tail, the head stays the tail. You have this positive autoregulation memory, but very few of these motifs which are noisy. In an upcoming lecture, we'll talk about oscillators, how there are clocks in biology, very important clocks. We'll talk about how they're built. And then you asked about cancer. So in diseases, what goes wrong, right? So this is a very important question. So a lot of times what happens in cancer, and maybe I'll use that also to set up what we're going to talk about today, which is signal transduction. What happens in cancer, for example, a lot of times is a mutation that happens in a gene. Mutation is a change in the gene, change of a letter or a little piece falls off or something like that. And these mutations, a lot of times don't do anything. A lot of times they kill the cell. But sometimes they don't kill the cell. They make a change in its brain, making it think, I'll tell you in a minute, that it's supposed to divide when it shouldn't. So we'll talk about. In fact, you need a few of those mutations in order to make a cancer cell. You need to be able to divide and ignore the fact that there are other cells around, and call in new blood vessels, and tell the immune system, don't eat me, even though I'm behaving in not a very social way to the other tissues, right? All these, you need a few mutations. And then you get a cell that is acting in a kind of, you can say, selfish way, or maybe recreating embryonic programs of making new tissues, but messy kind of tissues inside where it shouldn't. You know, that's a vulnerability of multicellular organisms. We have that program inside every cell, how to make a new tissue, and it can be activated by mutations. And then it's the field of cancer biology in the last, let's say, 4 decades, has mapped thousands of possible mutations and errors like that. And then it's important to know because sometimes that error makes a protein that's different, and you can make a small molecule as a drug that repairs that in a certain sense. And that's cancer drugs, modern cancer drugs, biologic drugs, work on the knowledge of what mutation is there and how to repair it. So indeed in some pathological cases like cancer, we say that mutations are many times the origin of this cancer. Other diseases, it's not exactly mutations, it's something else. In a future lecture, we'll talk about, let's say, diabetes, which is a metabolic disorder, where that comes from. Different, but it's a breakdown of this logic of the cell. Did I answer your question a little bit? No. Oh, you want to say specifically?
It might be beneficial for a system like that to become oscillatory to.
Yeah. So So you say that maybe changing this plus to a minus can now make a new behavior that's not so good. So, I mean, I think in general principle, that's right. In practice, it's a little more complicated. But yes. Okay. So how does this cell, let's say cancer cells and normal cells, let's say know when to divide and when not to divide? Yeah.
Making a plus to a minus be an unreasonable mutation. I mean, making a plus or a minus go away, I can see, but turning a plus into a minus, I have no idea how a mutation can cause
Yeah,
that.
good point. So how can a mutation turn a plus arrow into a minus arrow in a transcription, right? Transcription
In this
sense, yeah.
context.
Yeah. So yeah, that's maybe some mutations are more common, others more rare, right? And a lot of times actually the cancer mutations are really rare. Like it's a specific change in p53, not like a deletion of the thing, because the common mutations, the cells have fail-self mechanisms against them. It's those rare mutations. But I think there are ways. Like for example, if you have a mutation that now makes your activator bind a co-repressor that didn't have before now, which is so strong that it overrides. That makes sense? Something like that. So this can happen. Yeah.
It sounds super rare.
Yeah, as I say, some mutations are common, some are rare, but so a lot of times those rare mutations are found in cancer, like specific hotspots. Yeah, and then you sequence a lot of cancer and you say, oh, this mutation in position 503, changing serine to threonine occurs in this patient, this patient, this patient. Wow! So it's a lot of times those rare mutations, we'll talk about that too. Why is it that the rare mutations are vulnerable and the common mutations, cells have ways to overcome the common mutations? That's another kind of robustness, great. So we're talking about in human cell, in human body, we start from a single egg, we reach 10 to the 14 cells, that's 10 to the 14 divisions. And we keep replacing our cells, right, once per month, more or less, different tissues. So we make 2 million blood cells a second, red blood cells a second, for example. A lot of divisions. So we got to face the naturally occurring variation of mutations, right? Our cells need to be robust with respect to mutations. Otherwise, we get cancer right away. Okay? But mice, by the way, are designed to live about 2 years, and they get a lot of cancer after 2 years, lymphomas, blah, blah. They die of cancer. We start getting cancer usually at old age. Unless we're born, by the way, with one of those mutations. If we're already born with one of those mutations, we get cancer at an early age. That's if we're already born with it. It's not enough, but then a second mutation. Am I making sense here? It's a very interesting discussion, yeah. So that's another kind of robustness in multicellular organisms, is robustness to mutations that I'll describe, let's say, activate signaling pathways. Or we'll talk about how we'll talk in another lecture how cells can or how tissues can be robust to the most common kind of worst kind of mutations. That's very interesting. Only discovered 2016. All right, so we're talking about robustness and how important it is. So we're going to talk about in this class about something a level of regulation that we've so far treated as instantaneous. Okay, so far we had a signal Sx, activate transcription factor X, right? It became active, went to the gene, bound it. This is transcription. And sometimes this Sx is a small molecule, like a sugar that binds X, which is very easy to understand. But sometimes there's a whole world inside here, which happens on the time scale of seconds to minutes. And that's called signal transduction, especially when the cells need to move information from the outside world into the cell and respond. For example, the presence of other cells. Is there space around me? Right? I need to sense something from the outside and move it inside. Or, what's the osmotic pressure? What's the osmolarity outside? If it's too high, I need to build something inside in order to balance it. If it's too low, I need to cool down, like to release osmotic pressure so I don't burst, right? So this is called moving information from the outside to the inside. It's called signal transduction. And it works very fast. And it culminates a lot of times in the activity of a transcription factor, or sometimes in other machines that do something to the cell. Okay. Not always gene transcription, sometimes other kind of responses. So we're talking about signal transduction here, and signal transduction. from the outside to the inside. And the way this works, this is the cell membrane, right, the wall of the cell. And the first step is a receptor, which is a sensor that's half outside and half inside, usually. And this senses our signal. So for example, in the cancer case, cells in our body, in order to divide, they're very well behaved. They need a lineage-specific growth factor, a hormone, like epidermal growth factor for the epithelial cells and HGF for the liver cells. They need permission in the signal. So the presence of this hormone, this is a small molecule. How is it detected? It binds like a lock and key to a receptor, specific receptor on those cells that they have a need to know, right? It's on a need to know basis. And this receptor is built so when it binds the signal, it changes conformation, and inside, this protein changes shape and now can catalyze a chemical reaction. And typically there's another messenger protein which is free to diffuse inside the cell. This one is stuck at the cell wall, right? Yeah. That is chemically modified. When it's chemically modified, it can diffuse inside the cell, let's say go to a gene, binds its site, and transcribe proteins that respond to the signal. For example, in the case of this cell division, you sense the cell division, the hormone, and eventually you express proteins. You translate proteins that make the cell start to replicate its DNA, double its biomass, arrange the chromosomes in a row, make these fibers that connect them, make sure that you have each pair of chromosomes aligned. And when they're all aligned, there's an AND gate over all the chromosomes. They separate, and you build a new cell wall, and you have two cells. So that cascade is instigated by this fast signal transduction network. Now, when the signal goes away, you want to stop. So you have these proteins that remove the modification. So when I say modification, you notice that in this step here, there's no new proteins made. Oh, I said here Z, right? It's not the same Z. So I'm going to call these Z1, Z2, Z3. No, not necessarily. I mean, it could be a negative feedback where you make your own demodifying enzyme. No new proteins are made here. This all happens on the scale of seconds. So it's chemical modification, for example. You add, so the most famous one is called, these are long words for those of you who don't know them, phosphorylation, which means adding a phosphoryl group, which is a charged group, and when you add it to Y in a specific place, it causes it to change conformation. Now become an active transcription factor. Go to the nucleus, bind the DNA, make new genes, make new proteins. And this protein here that removes it is called phosphatase. What it does, it takes this phosphoryl group and dumps it into the water in the form of inorganic phosphate. Where does the phosphoryl group come from? Usually you take it from ATP and you make ADP. These are all now little details. And just to show that mass is conserved, you take the phosphoryl from here, you dump it out in here, this becomes active. All this takes seconds. So it's like a small signal transduction reaction for you. So how many of you have heard the word phosphorylation before? Great, half of you. Yeah, and half of you, it's new. We have to get used to it. It's a long word. But sometimes, many times, you have receptor and Y0 becomes Y-phosphate, and the receptor phosphorylates in another place. You have phosphate, and you have phosphatases, and this is now not a transcription factor, but in fact a protein that can phosphorylate another messenger twice, let's say, and then this one phosphorylates another messenger. twice. And this one goes to the nucleus. And this one finds the DNA, let's say. And with all these cofactors regulates genes. So you have these cascades of phosphorylation. And this, what I described to you now, is, for example, the MAP kinase cascade, where this growth factor signal ends up in cell division. And in cancer, a lot of times what happens is a mutation in the gene that encodes for X makes it act. Let's look at this. It makes it locked into its active conformation even without the signal. That's a mutation. Okay? And that means that pathway is saying divide, 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 even though there's no signal around. Okay? But cells are smart. They know that if there's divide, 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 they kill themselves, basically, because that must be an error. So there's another mutation that inactivates this killing themselves thing. So that's about cancer. And they lose their dependence on this growth factor. So cancer biology deals with signal transduction networks a lot, because also these receptors are fantastic drug targets because they're on the outside. They're on the outside of the cell. So you can make these small molecules that compete with the signals or that repair the. So there's here in Weizmann was discovered a way to make antibodies against these EGF receptors, and those antibodies help breast cancer, because breast cancer a lot of times has this mutation of this divide, 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 and these antibodies are like drugs that from the outside of the cell inactivate those mutant receptors. Isn't that smart? So that's like, unfortunately, after a while, a lot of times cancer finds a way to overcome, and some cancer cells are resistant to this, and that's a big field right now, how to deal with this resistance that happens to drugs. But okay, so that's like just an overview. Thanks for the question. It's a nice way to introduce signal transduction I didn't think of. But definitely a lot of biology, like in my Wolfson building where I work, is devoted to understanding signal transduction networks in great detail, because they have amazing detail, biochemical detail. We'll discuss a little bit of that, which gives them computational abilities that are crucial and not really well understood yet. So let's see. So we talk about precision in the face of noise, signal transduction, receptor, messenger, modification, phosphorylation. Whew! We talked about cancer. I think we're ready for a nice deep sigh of relief. Whew, you can make a ridiculous sound too. It's good. Okay, so let's talk about robustness, and in a way, also you can also say why do you need all these details? Like why double phosphor? Like why all these details? Sometimes I ask myself, why not just make a simple? So a lot of times the reason is robustness, that you have details that on their own make no sense, but together give you this amazing mathematical property to the circuit of being insensitive to, let's say, the concentration. So you see, there's concentrations of these receptors. So in you say human cells, okay, these growth factor receptors, you might have, let's say, 50,000 on the cell surface. But at 50,000, one cell will have 10,000, another cell will have 200,000. That's the way it is. Okay, it's huge variation. And then you have these proteins inside that can also be, tens of thousands of each other, thousands, tens of thousands. So the concentrations of the proteins in this pathway vary quite a lot between cell to cell. And still you want to not make mistakes. Sometimes you want to be very precise. How can you do that? So we need to talk about the input-output relationship. We're going to talk about the input-output relationship, which means how the concentration of the signal translates into the concentration of this active transcription factor. That's going to be our question. This is the input-output relationship. So I'm going to say it again. How the information from the outside, S, translates into the concentration of this active transcription factor that translates genes. So how this information is passed. This is going to be the input-output function of the signal transduction network. So just to make things very, very well defined, hopefully. we're going to have the input, quite generally, S, the output, some function of S, and this is it. This is the input-output relationship. And now, this is I calculated this with some values of the proteins in the circuit. Now I can ask, what happens if I change it? So this is in one cell. Now I change it to another cell. What happens to this curve? Yeah? So if this curve, so this is cell number 1, right? If this curve changes when I change protein levels, this is cell number 2, I call this a non-robust input-output curve. Why do I call it a non-robust input-output curve? Because for the same input signal, suppose this is a signal, for the same signal, the two cells have different output, different responses. Did I explain myself? That's a non-robust situation. A robust situation would be, yeah.
In this process, it's X, Y, Z, S?
No, S is the external.
Or S, X, Y, Z? What is the process? Like, is there a cascade in it?
Yeah, so I'm going to soon define the process. So if you think about this one, I have the receptor level, the level of Y, the level of Z. In this case, there's 3 proteins in the circuit. Do you agree? So in one cell, there's, let's say, 1,000 of these, 500 of these, and 200 of these. And I calculate I give some signal, I calculate. Now, I give all possible levels of signal and calculate the output. Now there's another cell. It has a different amount of X with it. I get another function. Thanks for asking. Was I clear here? No. So the signal transduction pathway works on a level of seconds. Cells have a certain amount of protein, and that lasts an entire generation. So cell number 1 has X, Y, Z, 1,000 receptors, and cell number 2 has 500 receptors. Now, for cell number 1, I test all possible levels of the input signal, and I measure the output YP, and I get a curve. Cell number 2, I do the same. Those curves are different. I call it a non-robust input-output relationship, because for a given level of signal, those cells will show different responses. Same cell type. Let's say even genetically identical cells sitting right next to each other on the same microscope slide, but they still have this noise in protein levels. So it's really important that you ask, so we're all on the same page. No, okay. Cell number 1, as I say, 1,000, 500, 200. And cell number 2 has 500, 800, 100. And now I take, try at all levels of S. And measure the output. Let's say YP. And I get a curve for cell number 1, another curve for cell number 2. And if they're different, I call it non-robust. And of course, if they're not different, I call it robust. So if all cells, despite having different levels of proteins, all respond to a signal the same, that's to say they learned how to cancel out their differences in the number of receptors, then I call it a robust input-output relationship. Yeah?
Don't we think about robustness as the cell is continuing to live even though things happen with different signals? I mean, if on cell 1 and cell 2, even though they have different ways, they will continue to describe.
Yeah. So the question is, isn't robustness continuing to live? So in a certain way to say, if the function is living, this definitely cells are very robust. And I drew here in the beginning an oak tree, which is a symbol of great robustness to many difficulties. Yeah. And here I want to talk about a specific function. So that's built of robustness in many, many, many steps. And I want to hear about a function of a humble little signal transduction circuit with respect to protein variations. And a lot of things need to be robust in order for cells to survive. Did I explain myself? Now look, not all signaling circuits are robust or need to be robust, because it's good sometimes to have cell-to-cell variation. So to be unpredictable. And indeed, a lot of signaling circuits, like with this growth factor, some cells divide, some don't. There's variation. But sometimes it's critical to have precision. For example, if you are sensing osmolarity of the medium, you want to know what's going on outside, and you want to adjust your osmolarity inside so you don't burst or shrink, you need a very precise reading. There's not a lot of room for error. So in some systems, precision is important in order for the cell to live in different conditions. In other systems, variability is fine. And this noise and protein levels generates variability.
So will this count as robustness?
This will be robustness of the input-output relation with respect to protein variation. So it's robustness of this with respect to this. There's a lot of kinds of robustness. Robustness of cells not to become cancerous in the face of mutations. It's a different kind of robustness. Big animals like elephants have a lot more cell divisions than we do, and they don't get cancer. Or whales. And it turns out that they have, for example, in places where we have one sensor of DNA damage, it's p53, one gene, they have 20 copies of it, for example. So they're evolved to be, because of their situation, of huge mass, evolved to have more stringent checkpoints on DNA damage. That's just for example. So, okay, I'm going to assume we understand what we're looking for. We're looking for, can a signaling circuit be robust to protein levels? And I want to tell you that this is not so easy. And up to about 10 years ago, I didn't believe that it's possible, because you're talking about a dynamical system that has parameters, x, y, z, and now you want its behavior to be invariant to these parameters, completely invariant to these parameters. That's very rare, unusual. Okay? Of course, evolution can reach the unusual place. That's the beauty of biology. But just to demonstrate it, let's solve a circuit like this.
But we're not talking about robustness to any level X, Y, Z. We're talking about levels which exist in cells.
Good point. We're talking about the range of variation in cells. So there could be, we'll talk about it later, if there's no X, you can't be, if no receptor, there's no signal. Absolutely. So there's limits to it. Exactly. So the naturally occurring variation.
The robustness itself needs to be defined in some way, right?
Yeah.
It's not well defined.
Right. So let's say, when I say variation protein level, I can say I want the input-output curve to vary less than 5%. If proteins vary tenfold, let's say, something like that. Something like that is even more quantitative. That's how engineers define it. Yeah. 6 sigma, 6 standard deviation, a variation in components. Any more questions now? I think maybe it's still not clear. Yeah.
When you're talking about variation, say the amount of receptors,
Yeah.
is there usually like precision in the distribution? When you look at the variation, you see distribution. Is this distribution usually the same for copies of the population? Like it can be a parameter that the cells use.
Okay, so I guess if I understood your question, if we look at different cells and we ask what's the probability of X, so how many times the cell will have 1,500, that gives a probability distribution function of X between cells. So a lot of cells have this value, fewer cells have this value. These probability distributions have typical shapes. Okay, and when you measure them, they look like gamma distributions. So sometimes they look like this, sometimes they look like gamma distributions, and their mean and standard deviation vary from protein to protein. If there's negative autoregulation, for example, you get a more narrow distribution. and their standard deviation divided by the mean is a function of the mean. A lot of times look like this, and there's something like 10% variation in highly expressed proteins, and you can have, let's say, 50% variation in low expressed proteins, let's say around 100 copies per cell, and here let's say maybe 10,000 copies per cell. And this is measured, so that's kind of a universal curve. Of course, there's exceptions. Some proteins are more variable. Stress response proteins a lot of times, sometimes more variable than others, and so there's definitely patterns. Did I answer your question? Okay. This is kind of more for experts. And so input, output, non-robust, robust. So look, let's. I just want also to show you a little bit the math of these kind of reaction systems, where we have interactions between proteins. So we're going to solve this system. What we're going to solve is we have receptor X, and it takes protein Y and adds this phosphoryl group. This is a signal. The velocity, the rate this happens, we'll call V K of S. K is because this enzyme that adds phosphoryl group is called a kinase in general. It's a protein kinase. So the more signal there is, the more this reaction happens.
Can you define VK again?
It's the rate where X adds this phosphoryl group to Y. I'm going to write the equations in a second. And then we have this other protein Z here, which works at rate Vz. This is the, okay, let's call it Vz. Great. So this removes, it's a removal of the phosphoryl group from Y phosphate. So let's write down the equation for Y phosphate. This is the rate of change of Y phosphate. Now these are equations that describe basically collisions between proteins. So Y is floating in the cell in its form Y0, it's not phosphorylated, collides with X, and X, actually what it does is breaks an ATP and glues the phosphate onto Y. The probability that it does this is proportional to whether it binds a signal or not. The more it binds a signal, the more probability it has to do this. So the way we write down this kind of reaction is we say it goes like this. It goes like how much unphosphorylated Y there is times how much X there is. This times means this concentration of this times concentration of this is basically the probability that they collide. The more concentration of either, the more probability that they collide. Yeah. And here we put in this rate, because if they collide, this is the probability that they'll pass on the phosphoryl group. This is called, this kind of thinking is called mass action, which is the rate times the concentrations of the reactants in this chemical reaction. And what's the rate that Y phosphate goes back to Y0? So it's minus. Y phosphate is lost.
Is there never not enough ATP for this?
Oh, yeah. So ATP, let's put in ATP here. ATP here goes in, and ADP goes out because the phosphoryl goes out. So if the cell is really starving, there's not enough ATP, that's a problem. Cells are really good at maintaining, it's another kind of robustness. They retain their concentration of ATP quite well, especially ATP to ADP ratio, quite well with special robust circuits. So not to run out of energy. So we're going to assume there's enough energy, all right? So YP collides with this Z, and this is the rate where phosphoryls are removed. So we have this addition of phosphoryl to YP and removal, phosphorylation and dephosphorylation. Questions? Yeah.
Causes YP is the signal for the.
Yeah, this is our output. Output, what we called Y star, activates genes, that respond to our signal. Yeah.
So X is the number of receptors.
Yes.
Vk of S is the rate that X converts the receptors to an active form.
Yeah. It's basically activity. You can say activity per receptor. So this depends on the way the receptor is built. So each receptor has, the way it's built determines Vk of S. If the receptor is mutant, for example, like in cancer, Vk of S is high and independent of S. If it's a normal receptor, this Vk of S depends on how much S is bound, right? If S is bound, it's more probability to be in its active form. And this happens very quickly. So Vk of S is actually an average over many conformational changes that happen on the microsecond time scale, and bindings of S that happen on the millisecond time scale. And we get a number, which is, given the concentration of S, what's the probability that X will be in its active form? So if Y0 binds it, it'll transfer its phosphoryl group. It's a little bit complicated, I know, but I think you give it a chance and it'll become kind of a natural language, this mass action thinking of proteins here diffusing, colliding, moving modifications, yeah.
So VKS is dependent on the signal, but the second term is just dependent on the amount of YP?
This one?
Yeah.
The dephosphorylation signal? Yeah, so in this case, Z is floating inside the cell, doesn't know about the outside world, and when it finds a phosphorylated Z, a Y phosphate, and hits it, it has a certain probability to move the phosphoryl out into the water in the form of inorganic phosphate. And then something else takes care of that. Puts it back on ADP to make new ATP. Everything closes down. Well, we don't need to care about that. Yeah?
Shouldn't Y0 be dependent of YP? Because maybe they're active.
Okay, great. So we need one more equation, which is conservation of Y proteins. So Y0 plus YP together make up YT, total Y. So in this system, there's a certain amount of total Y protein, and some of it is Y0, some of it is YP. So our cell has a certain amount of X, a certain amount of Y, it's called Y total, a certain amount of Z, and this a certain amount of Y is partitioned between Y0 and Y phosphate. And our goal is to ask, is Y phosphate, okay, for a given signal level, sensitive or insensitive to those amounts of X, Y total, and Z? So we have two equations, equation 1 and equation 2, that we're going to solve right now. Yeah, I hear there's more questions. I just want to erase, so give myself time to collect myself. Maybe take a nice deep sigh of relief even. Just think about this when you teach, how many steps it's possible to skip, and so I'm very grateful for these questions. So keep asking. So who said, yeah?
Z is independent of time but varies between cells.
That's right. So Z, you're asking is Z independent of time. So here we're going to talk on the time scale of seconds. And Z is a protein level that stays constant. We remember how much. It has this, its dynamics are governed by the degradation dilemma. It stays constant for about a cell generation, which is hours, right? Hours or hour. So on time scale of seconds, we can assume X, Y, and Z are constant for this cell. So separation of time scales. And indeed, the cell has this individuality. It has X, Y, Z. It lasts for about a generation time. Yeah.
What did you write for Y0? I think
This thing?
No, mass action. I didn't.
This mass action?
Mass action rate.
This is a mass action equation for the collision of Y0 with X, causing Y0 to become phosphorylated. So this concentration of X times concentration, this is the collision probability, and this is the probability that if they collided, you have this.
So you didn't write this for Y0.
Yeah.
You wrote it for the
Thanks.
whole
Yeah.
Okay.
Thanks. Yeah.
Basically, Z doesn't have to be constant. It can be dependent on Y and it can be dependent on many things.
You say in principle Z can be dependent on many things. Correct. And here in this circuit we keep it constant. It's just a diffusing Z and a simple circuit so we can get the hang of it. Yeah.
So this might be a stupid question, but you measure concentration, for instance, in units of some number divided by volume or
Yeah.
whatever.
this clearly, the units are not balanced on both sides of the equation, right?
Okay, so let's say about units. The units are very important. So here we have, let's talk, it could be numbers or concentration. Let's talk about concentration. Thanks. Concentration, okay? So let's talk about units. I'm going to write down, so it's getting a little bit more, I'm going to use red here for the units. Here we have units of concentration. Okay. Here we have units of concentration times concentration. Oh, sorry, here we have units of concentration divided by time. This is rate of change. So this tells us what units of this thing is. The units of this thing has to be 1 over concentration over time. Or, just to make it intuitive, this means that this is the rate of phosphorylation per receptor. So this unit, and then, okay, and what about these units? Also, 1 over concentration over time, because it's the rate of dephosphorylation per Z. This makes sense? This will play an important role later, but it's important to say these are what's called bimolecular constants. They talk about the reaction of two molecules. So okay. Second order constants, yeah.
The first term happens only on the cell wall, right?
Yeah.
And this happens everywhere?
Yes.
So
maybe there's a correction.
yeah, I don't know. I mean, X may depend on the space.
So Itai is asking, receptors could depend on space only at the cell membrane, or maybe only at the one patch on the cell membrane, which happens to. So you can add spatial considerations here. Let's imagine the case where mixing is very fast. In bacteria, for example, this Z and Y can cross from side to side 1 micron in 10 milliseconds. That's the diffusion constant. So let's imagine everything is fast diffusion. Yes, and spatially homogeneous. In fact, there's a lot of approximations here, because sometimes in the cell, proteins maybe stick to little fibers and they can't really diffuse and localize. And so there's a lot of complications you can make here. But we're not going to, because we want to get the principles, right? So.
But VK of S actually serves a couple of roles here, right? Because it's also it's the conversion to active form based on the signal, the rate of phosphorylating, and also correcting for the units of Y0 and X and their interaction, right?
Yes. Yeah, there's a lot of physics going into this VK, including on rates and off rates and stuff. So you can make a more detailed model if you want to. But this is you don't want to. This is a level of approximation that's good for us. Yes, good. Of course, in this course, we're trying to make a balance between absolute detail and simplicity for understanding. So I try to choose the right level, which is kind of an art in itself. And sometimes maybe I get it wrong. So. All right, so here we have two equations and we're going to solve them. Okay, we're going to solve them. And the solution, we're going to solve them at steady state. So, if we add some signal at a certain concentration and wait a few seconds, everything comes to a balance. Protein levels haven't changed yet because they take hours to change. So at steady state, dyp dt, there's no change anymore, which means this equals to 0, this minus this equals 0, which means that phosphorylation equals dephosphorylation. This equals this. So Vk y0 x equals Vz yp z, which means adding phosphate balances removing phosphate. Phosphorylation balances dephosphorylation. Questions? Now I want to solve for y phosphate, so I want to get rid of this y0. How do I get rid of this y0? By noticing the y0 is y total minus y phosphate.